Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're investigating the effect of enzyme and substrate concentrations on the initial rate of reactions. So the rate of an enzyme control reaction is influenced by different factors: the temperature, pH, concentration of the substrate, and the concentration of the enzyme. When measuring the influence of one variable, all other variables must be in control. So to conduct this experiment, you need milk powder solution, trypsin solution, test tubes, test tube holder, stop clock, a 5 centimeter cube pipette, goggles, colorimeter, cuvettes, and distilled water. Method. So dilute the solution of trypsin as needed with distilled water to produce a range of concentrations such as 0.2%, 0.4%, 0.6%, and 0.8%. Use a control solution by adding 2 centimeter cubed of trypsin solution and 2 centimeter cubed of distilled water. This is set to 0 on the colorimeter. In another cuvette, add 2 centimeter cubed of milk suspension and 2 centimeter cubed of the stock trypsin solution. Place this cuvette in the colorimeter and measure absorbance at 15 intervals for 5 minutes. Rinse the cuvette with distilled water and repeat this with all the other trypsin concentrations. The risk assessment of this practical. So there's the risk of broken glass, there's a risk of cuts from sharp objects, so be cautious and keep away glass from the edge of desks. In case of emergency, elevate cuts, apply pressure, and do not remove glass from the wound, and seek further medical assistance. So there's the other risk of hot liquids. So there is the risk of scalding from hot liquids. Be cautious and use tongs to remove boiling tubes and wear goggles to minimize the risk of hot liquid splashing in your eye. In case of an emergency, run the affected area under cold water and seek further medical assistance. You also have the risk of enzymes, there are chances of developing allergies. Hence, it is best to avoid contact with skin and eyes by wearing goggles. In case of an emergency, seek medical assistance. So when you do collect the results from your practical, you need to plot a graph with the rate of reaction on the y-axis and the temperature on the x-axis. So what you will see in your results, what you will observe from your results is that milk contains a protein called casein. When casein is broken down, the milk goes from white to colorless. Trypsin is a protease enzyme which hydrolyzes the casein. Hence, as the concentration of trypsin increases, the rate of reaction increases as a result of the increase in the number of enzyme-substrate complexes forming. The increase in the rate of reaction eventually levels off, and this point is a saturation point as the enzyme is no longer the limiting factor. You can change the experiment such that you measure the effect of the substrate concentration instead by controlling the concentration of trypsin and using a range of different concentrations of milk. So that is all from Professor SNAB today. I hope you found this video useful. If you have any questions, let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share, and subscribe.